Welcome to the deep dive. We often think about natural selection in pretty simple terms, like survival of the fittest, almost as if any change is possible.
Right, like evolution just has a completely blank slate to work with.
Exactly. But when you really dig into the mechanics, like in the material we're covering today from Strickberger's Evolution, it becomes clear selection isn't working in this wide open space. It's more like it's well channeled.
Uh-huh. And that tension, that's really the core of it, isn't it? The power of selection hitting up against the organism's own internal rules and history.
So what's the mission for us today then?
I think it's to move beyond just adaptation and see how evolution actually manages change. We need to explore these dynamics. Selection, sure, but also genetic variation, polymorphism, these developmental limits, constraints, and canalization, and how fitness actually plays out.
Makes sense. The sources really emphasize that selection acts on the phenotype, what we actually see.
Yeah, but that action is always filtered through the genes underneath and, critically, these kind of rigid pathways of how an organism gets built. It's almost like a biological negotiation.
Okay, so let's start with those outside pressures. It's a key point that selection on one species never happens in a vacuum.
Definitely not. Everything is connected. Competition, predators and prey, even those cooperative setups like mimicry. It's like a big evolutionary dance involving everyone.
And this leads to the idea that everyone has to keep improving, right?
Right. Lee Van Valen put a name to that back in '76. The Red Queen hypothesis.
Ah, the famous one from Through the Looking Glass.
Exactly. It takes all the running you can do to keep in the same place. It perfectly captures that co-evolutionary pressure.
Because you're not just adapting to, say, the climate changing,
Yeah.
you're running against every other species that's also getting better.
Precisely. If species A gets an edge, its fitness goes up. But that automatically makes things harder for species B, lowering its fitness.
Which forces species B to adapt in response.
Exactly. It kicks off this constant arms race. And Darwin saw this way back. He wrote in Origin of Species that if some species improve, others have to improve too, or they risk going extinct.
So selection isn't just a one-off event when the environment shifts, it's continuous, relative.
Absolutely. Always running.
Like, speaking of running, let's switch gears from that external arms race to the internal rules. If mutations are always throwing up new variations, why isn't evolution just limitless? Why do we see these incredibly stable anatomical patterns?
That's the million-dollar question, and it brings us to developmental constraints. Think of the developmental system as the factory that builds the organism. It allows for some flexibility, some plasticity.
Which we'll get to.
Right. But it also imposes pretty strict limits. The research makes it clear. Really big developmental changes often just don't work. They're lethal.
And not because the change itself is inherently bad.
Not necessarily. It's more that the change is so disruptive, it can't be integrated properly with everything else that's going on in the embryo. These systems are complex, deeply interconnected, honed over ages. A sudden massive change tends to break the machine.
So the organism's own evolutionary history kind of limits its future options.
In a way, yes. That's phylogenetic constraint.
And the classic example of that is
The neck vertebrae in mammals. It's amazing, really. Almost all mammals have exactly 7 cervical vertebrae.
doesn't matter if it's a whale with a practically non-existent neck or a giraffe with that incredibly long one.
Nope. 7. Think about figure 15.1 in the text, showing the whale and giraffe skeletons side by side. Visual, it's striking. Same number. The point isn't the number 7 itself, but what it implies about the cost of changing it.
What kind of cost?
Probably huge pleiotropic effects. Changing that fundamental number likely messes up all sorts of other critical developmental processes downstream. It's just too integrated.
But it's not an absolute rule, is it? The constraint isn't total.
Good point. No, it isn't. The sources mention exceptions. The three-toed sloth has 8 or 9, the manatee only 6. So change can happen, but it's rare. Constraint narrows the path, but doesn't always block it completely.
Okay, and this ties into the idea of evolvability.
Yeah, it does. Compare frogs and placental mammals. Frogs have been around much longer, about 200 million years, over 3,000 species, sure, but they're all basically variations on the frog theme, stuck in one group, the Anura. Now look at placental mammals. They showed up much later, maybe 90 million years ago. Around 4,300 species, so a similar number. But look at the diversity. Bats, whales, primates, rodents. They exploded into 18 different orders, completely different body plans.
Wow, that's a huge difference in the potential to generate novelty.
Exactly. It shows fundamental lineage-specific differences in evolvability. past selection can also act as a constraint through trade-offs.
Ah, the master of one trade versus jack of all trades idea.
Precisely. If you become incredibly specialized for one thing, it often compromises your ability to do something else well. Think about a crocodile. Okay, great swimmer, right? Selection pushed for swimming efficiency, but the adaptations that make it a great swimmer likely hinder its ability to be a fast, efficient runner on land. Specialization comes at a cost, limiting future pathways.
So I've got these constraints, these limits, but organisms aren't totally rigid. There's flexibility too.
Right, and that's phenotypic plasticity. It's the ability of one genotype, one set of genes, to produce different phenotypes, different observable traits, depending on the environment.
How do scientists visualize or measure that?
They use something called a reaction norm. Basically, you plot the range of phenotypes an organism expresses against some environmental factor, like temperature, or maybe how many predators are around.
Okay.
If you get a single flat line on that graph, it means the trait is pretty fixed, regardless of the environment. That's called being highly canalized.
Buffered against change.
Exactly. But if the line slopes, or if you have different lines for different genotypes, that indicates plasticity. The phenotype changes in response to the environment.
The book gives the stone chat example, the Saxicola species.
Yeah, figure 15.3 shows this well. The Siberian stone chat lives where the seasons are very predictable. Its reaction norm for when it starts its molt is very narrow, very fixed.
Makes sense. It doesn't need to adjust.
Right. But the African or European stone chats live in less predictable climates. Their reaction norms are much broader, showing greater plasticity. They can adjust their molt timing more depending on the conditions they experience.
And this brings us to canalizing selection. What's that?
It's basically selection that reduces plasticity. Waddington and Robertson describe this. It favors genotypes that produce a consistent, reliable phenotype, despite environmental fluctuations or even genetic variations. It sort of tightens the response range.
Okay, interesting. Now, the next concept, genetic assimilation. This one sounds tricky. Almost Lamarckian, like inheriting acquired traits.
It sounds like it, but it's absolutely not Lamarckian. That's a crucial distinction. It's pure Darwinian selection act on existing, maybe hidden genetic variation.
Okay, break that down. How does it work?
Think of it like this. An environmental trigger, say, a heat shock, causes a particular phenotype to appear in some individuals. This phenotype was always possible genetically, but it needed the environment to reveal it.
Okay, the environment unlocks something.
Right. Now, if that environmentally triggered phenotype is advantageous, selection can then favor the underlying genetic variations that make it easier to produce that phenotype. Over generations, selection can refine the genetic basis so much that the trait starts appearing even without the original environmental trigger.
Ah, I see. So the trait becomes genetically locked in or assimilated.
Exactly. The potential was always there, hidden in the genetic variation, the environment just exposed it, and then selection fixed it.
The sources mention experiments showing this.
Yes, classic examples. Waddington's work with Drosophila, where an ether treatment could induce an extra pair of wings due to a mutation, Ultrabithorax. After selecting for flies that showed this trait, he eventually got flies that developed the extra wings without the ether treatment. And another is the color change in the tomato hornworm, Manduca. A heat shock could induce a certain color form. By selecting for individuals that showed this response, researchers bred a line that produced that color more reliably, even without the heat shock. Genetic assimilation in action.
Okay, let's shift to the outcomes of selection. How do we actually measure fitness?
Fundamentally, fitness is just reproductive success. How many descendants does one phenotype leave compared to others in the population? That's the bottom line.
And we can quantify the strength of selection.
We can, using the selection coefficient usually symbolized as S. We typically set the fitness of the most successful phenotype to 1. Then, the fitness of another phenotype is expressed as 1 minus S.
So if S is, say, 0.1.
That phenotype has 10% lower reproductive success than the fittest one. A higher S means stronger selection against that phenotype.
Got it. Now, selection doesn't always just eliminate variation. Sometimes it actively maintains it.
Right. That happens through heterozygous advantage, also known as heterosis, sometimes called hybrid vigor.
This is where the individual with two different alleles, the heterozygote, is actually fitter than either homozygote, the individuals with two identical alleles.
Exactly. And this has had huge practical impacts. Look at hybrid corn.
The yield increases are legendary.
Absolutely. Back in the 1920s, yields were maybe 25 bushels per acre. By exploiting heterosis through selective breeding, developing hybrid lines, yields shot up. Now they can be 140 bushels per acre or even more. That's the power of heterozygous advantage.
And this mechanism maintains different alleles in the population. That's balanced polymorphism.
Precisely. A term coined by E.B. Ford. It refers to selection actively preserving genetic variation. For it to count as polymorphism, the second most common allele generally needs to be at a frequency of at least 1%.
The classic textbook example must be sickle cell anemia.
It is. The HPS allele causes sickle cell disease in homozygotes, HPS, which is often lethal, so ZEC is very high against them. But the heterozygotes, HPS, have a significant advantage in areas where falciparum malaria is common. They are much more resistant to severe malaria than the HPA homozygotes.
So the heterozygote advantage keeps the dangerous HBS allele circulating in those populations. Figure 15.4 in the text maps this out geographically, showing the overlap.
Exactly. And it's another arms race, too. The malaria parasite, Plasmodium falciparum, is constantly evolving itself, developing genes to try and get around our immune defenses. It's relentless coevolution.
Now, this idea that selection maintains so much variation wasn't universally accepted. There was a major alternative view.
Right. Motoo Kimura's neutralist hypothesis, which gained traction in the late 70s and 80s.
What was his argument?
Kimura argued that most molecular evolution, especially changes in amino acids within proteins, is actually selectively neutral, meaning it has no real effect on the phenotype's fitness.
So how would those changes become common?
Primarily through genetic drift, just random chance fluctuations in allele frequencies from one generation to the next. He argued the cost of selection required to maintain all the observed polymorphism via mechanisms like heterozygote advantage would just be too high for populations to bear.
Interesting. But the evidence presented here seems to push back against a purely neutral view.
It does. Several lines of evidence support a strong role for selection. For instance, you see clear correlations between the amount of allozyme polymorphism, variation in enzymes, and how variable the environment is. More variable environment, often more polymorphism,
Okay.
and maybe even more compelling. If you compare polymorphism rates in different parts of genes, you find much higher rates in non-coding regions, like introns, compared to the coding regions, the exons, that actually specify the protein sequence.
What does that imply?
It implies that changes in the exons, the functionally critical parts, are often harmful and are actively removed by selection. Changes in introns, which often don't affect the final protein, are tolerated more. This difference strongly suggests selection is scrutinizing those coding sequences. It's not all just random drift.
Okay, let's wrap up with some really visual, striking examples of selection you can practically see happening. Mimicry is a great starting point.
Absolutely. Henry Bates described Batesian mimicry first. This is where a perfectly edible species evolved to look like a toxic or unpalatable species, the model.
So predators avoid the mimic because they've learned to avoid the nasty model.
Exactly. But there's a catch. It works best when the mimic is less common than the model. If there are too many mimics around, predators might start attacking them, realize they're okay to eat, and the protection breaks down. It's frequency dependent.
The monarch and viceroy butterfly example is classic here, right? Figure 15.6a shows them. Monarchs are nasty, viceroys copy them.
Right, and this isn't a new trick. The text mentions an amazing fossil, Eophilium mesalensis, a stick insect from the Eocene 47 million years ago that looks incredibly like a leaf. Mimicry is an ancient strategy.
And then there's Müllerian mimicry. How's that different?
In Müllerian mimicry, multiple unpalatable species converge on a similar warning pattern. Think of various species of wasps or bees with yellow and black stripes, like in figure 15.6b.
So they're all genuinely dangerous.
Yes, and they all benefit because a predator only needs one bad experience with any species in the group to learn to avoid all species displaying that pattern. It's like a shared advertising campaign for "Don't eat me."
Very efficient. Okay, now for maybe the most famous example of rapid observable evolution driven by environmental change. Industrial melanism.
Yeah, the British peppered moth, Biston bechellaria. This happened in what the researchers call a coarse-grained environment, meaning that within the same general area, different individuals could experience drastically different environments and thus different selection pressures. In this case, landing on a soot-covered tree versus a clean lichen-covered one.
Right. Before the Industrial Revolution, the typical peppery white moths were well camouflaged on lichen-covered trees.
But then industrial pollution killed the lichens and blackened the tree bark with soot, especially near cities. Suddenly, the rare, dark, melanic form of the moth had the advantage. They were beautifully camouflaged against the dark background. You can see this in figure 15.7.
And the pale ones stuck out like sore thumbs to bird predators.
Exactly. Bernard Kettlewell did those crucial experiments in the 1950s. He released marked moths of both types in polluted and unpolluted woodlands and then recaptured them.
What did he find?
The results were dramatic. In polluted woods, far more dark moths survived to be recaptured. In clean woods, far more light moths survived. Clear evidence of strong selection by predators.
And the change in the population was incredibly fast, wasn't it?
Astonishingly fast. The frequency of the dark form went from very rare to dominant in industrial areas in maybe 50-100 years. Calculations suggest the selection coefficient, SS, against the non-favored morph was really high, maybe 0.2 or even more in some places. That means a 20% or greater survival disadvantage.
That's intense selection.
It drove the frequency of the typical form down from like 98% to just 5% in about 40 generations in the heavily polluted areas. A powerful demonstration.
Is there a modern equivalent people are studying?
Yes. The rock pocket mouse, Catodipus intermedius, found in the American Southwest, often living on differently colored rocks.
Like dark lava flows next to light sandy areas.
Precisely. And guess what? The mice living on the dark lava flows are predominantly dark-colored, while those on the light sand are light-colored, as shown in figure 15.8. It's camouflage again, driven by visual predators like owls.
And do we know the genetic basis for this one?
We do. It involves mutations in a key gene controlling pigment production, the MC1R gene. What's really neat is that in different populations of these mice living on different lava flows, slightly different mutations in the same MC1R gene have caused the dark coloration.
So evolution found similar solutions via slightly different paths in the same gene.
Exactly. And crucially, the difference between the light and dark forms often comes down to just a few, maybe only 4 amino acid changes in that MC1R protein. It shows that significant adaptive phenotypic changes can result from relatively small genetic tweaks when selection pressure is strong. #outro. So pulling it all together, what this deep dive really shows is that evolution isn't just this, free-for-all adaptation process. It's a constant negotiation.
Negotiation between
The external pressures from the environment and other species, the internal limits imposed by how organisms are built, those developmental constraints, and then the actual raw material available, the genetic variation, the polymorphism, and the flexibility offered by plasticity.
It's the interplay of all those factors that really channels the path evolution takes.
Definitely. The final outcome, the phenotype we see, is a product of all those forces acting together.
Okay, so for a final thought to leave our listeners with, let's circle back to that rock pocket mouse and the MQ1R gene. The material also mentioned pale lizards evolving on white sand dunes in New Mexico.
Right. Another case of camouflage adaptation. And again, linked to the MQ1R gene.
But here's the twist they pointed out. In one lizard species, the pale color came from a mutation affecting how the MQ1R receptor sends signals. In another species on the same dunes, the pale color came from a different mutation in MQ1R that affected how the receptor protein sits in the cell membrane.
That's fascinating. So phenotypic convergence, they both ended up pale.
Yes, same adaptive endpoint.
And genetic convergence, in a sense, mutations in the same gene, MC1R.
But mechanistic divergence, the specific way the gene mutation caused the paleness, was different.
Wow.
So the question for you to ponder is this. If you can arrive at the same adaptation, pale color, via different functional changes within the same gene, what does that tell us about how predictable evolution really is? Is the path fixed, or is it more contingent on history and chance, even when the destination seems the same?
That really highlights the complexity, doesn't it? It suggests the evolutionary channel might have multiple routes, multiple ways to solve the same problem, even at the molecular level.
Something to definitely think about. Thank you for joining us on this deep dive, exploring the channels and constraints that shape the course of evolution.